Hi everyone. I am Louisa Ho. I manage the commercialization technologies coming out of Wilfrid Laurier University. And welcome to the Phytotoxicological Device Breakout Room. If you are in the wrong room, you can enter another breakout room if button at the bottom, or you can stick around for a bit and check out other breakout rooms throughout the session. So I want to make all of you aware that the session will be recorded. And first, I thought it would be a good idea to go over a little bit of the commercial side of the technology. So I will do that now. Okay. So can everyone see my screen? Okay.
Yep.
As Kevin mentioned, this is his device in panel one. That's the overall design, and he has two different designs of cartridges. These are covered by a patent that you can look up. It's public now by just Googling the patent number here. And if you have any questions about the technology as you're preparing your abstract, you can direct them to me, and then I will be able to answer them or have Kevin answer them for me. The technology advantages that Kevin highlighted, but can also be added based on the commercial products that are out there, is that it's simple and cost effective. It has a rapid assessment compared to lab-based tests that are the standard right now. So it can be done between 1 to 3 weeks instead of months. It's portable, it's field deployable, and it allows for site-specific assessment, which many of the current practices don't. It's continuous flow, so it has realistic exposure conditions. So you can see over time or at the specific site, and different timelines depending on when contaminants might be exposed during different times of the year, such as if there's a melt and allowing water flows to change during different seasons. And it allows for concurrent chemical analysis to take place as well, if you can sample the plant tissues that are grown using the device. And finally, there is a potential for robust analysis that we think is better than the alternative that will allow regulators and others to have better decision-making abilities. So I also wanted to let all of you know that there was a market research study that was conducted back in 2013 on this specific device. And what they found was that a key customer segment that they thought we should target was the site-specific risk assessments of contaminated sites. And so the customers would be likely environmental consulting engineering firms. And so what they were looking for is a service to be able to conduct these field deployable assays for them. So the market opportunity is the service. Right now, ecotoxicological testing can go for about $100,000. These devices will allow the service to be as little as $5,000 per site. The annual market size based on the number of contaminated sites that need to be evaluated per year in North America is about 5 million. And competitive analysis shows that there are no direct competitors for this, and there are only a few rapid kit products on the market. So now I'll just open up the floor for questions. So feel free to put something in the chat box or to just yell out your questions.
Sorry, I was just writing everything down. I do have a question about the application. So I know that it's supposed to be used in sampling water as well as wetlands. I was wondering, could it just be placed in soil as well, for example, surrounding farmlands?
So we have a modification that I didn't show that would do seed germination assays in drier areas. So it does require that the water be able to move into the exposure chambers. So it has to be wet enough for water to move. But if it's wet enough for plants to grow, this modification that we have will allow us to conduct assays in upland areas.
That's
But my
great to
background
hear.
is I'm a wetland ecologist, so my focus has been on wetlands, so rivers, streams, lakes, that type of thing.
That absolutely makes sense, especially because I know in Canada, most of the bioremediation efforts for mining areas tends to be to make man-made wetlands now. So
Yeah, and even a lot of urban areas are serviced by stormwater management ponds, and it's unclear how the contaminants there are impacting things. So
that was actually going to be my next question was, have you considered doing man-made sites for water runoff, such as in urban areas or even in sewer systems?
yes, so yes to the first question. We're actually working with a firm out of um, Milton, looking at stormwater management ponds and how they function and contaminant effects. We've been working on this for maybe 2 years. The research has been building, and then we were really starting to take off and get some good studies underway. And then with COVID, we had to stop doing that, but we've resumed that now. So we are looking at um, urban areas and um, wastewater treatment plants is another area that we're very interested in because there are a lot of pharmaceuticals and personal care products that are discharged into our watersheds through wastewater treatment plants. So we're interested in looking at the effects of those too. And some of them are really, really fascinating.
And in some regions, I just want to add that during the winter, when the roads are salted, that also affects it's also one of the contaminants that you see in the yeah, in those regions.
Some of our stormwater management ponds, the water gets so high in salts that it's approaching brackish
Yeah,
levels, which is it's incredible to think about that. But they do approach those levels.
yeah, and it differs by municipality. Not all municipalities allow it, and some do.
That's a really good application, actually. In my undergrad, we had to measure trees throughout Guelph around the city to see when they started to change color and then eventually fall off. And any that were close to major roadways that are salted will experience that change in season a lot faster because of the stress.
So you're from Guelph?
I am from Guelph, yes.
What program did you go through that?
I did I did my undergrad in biology and a minor in biotechnology. I specialized in plant science specifically, though, and now
Oh,
I'm
really?
currently doing my master's in biotechnology.
Oh, I went to Guelph. Um, for at that time there was the botany department. So did you have Brian Husband for anything?
I did not know. We actually don't even have, from what I know, we don't even have a botany department anymore. It's plant
No, we don't.
agriculture took everything.
Yeah, yeah. Oh, great school.
Yeah, especially for plants.
Yeah,
Yes. So
yeah.
I think, Kevin, one of the challenges with commercializing this is having the available seeds for the sites that you were particularly looking at. So if it was something in Southern Ontario, then Laurie has a pretty good bank of seeds um, to cover that. But if there was, I don't know, and I think in Yellowknife, you probably took some as well. Um, so what are the challenges you think there might be for that, the different sites?
Well, I guess there's two ways that you could look at it. One would be to use the same species of plant in a number of different sites, so you get familiar with how that one particular plant grows and responds.
Mhm.
Um, and so that, that you could use a plant that, we call it a phytometer, a plant that may not be found everywhere, but you would use it in your assays because it's something that can be worked with. The opposite approach would be to collect seeds that are specific to a given region where you're interested in seeing if there's an effect, and actually looking at the effects on the plants that are native to that area. So there could be quite a, it could be quite a challenge to collect seeds of everything from everywhere, but we could start small, and we've got a pretty good collection at Laurier already. And every fall we go out and collect more seeds, and we can grow them in the greenhouse too. But I mean, that would, if this really took off, having a small nursery where you could obtain seeds would, I think, be a logical aspect or development to have.
Mm-hmm. Would there also need to be a level of standardization for like knowing how the plants respond to various contaminants?
Yeah, and I'm quite sure that you're going to find some plants, some species that are more sensitive to some contaminants than others, and so once that's we know that's the case for some things, but I could see developing a whole library of responses, and if you suspect a particular contaminant in an area, you could choose a set of species that are more sensitive to that to put out. So you could target the species to the contaminant, or if you just want to see if there's any general contamination, you might put out a range of species that have a range of sensitivities.
Mm-hmm.
So the whole approach is letting the plants speak for themselves rather than using a surrogate, which is really what the water sample is. The values that we use, our targets with these water quality parameters, are based on what we expect would be a level that won't affect a biological entity. So this is going a few steps back and letting the organisms speak for themselves.
I also wanted to mention that you had been playing around with the 3D printer on making these devices as well, and had come up with a pretty good prototype that might even be cheaper than the original designs. You want to speak a little bit about that?
So there is another design that we came up with, and it's specifically for, I got to be careful when I say this. So say drier areas, and so for someone who works in wetlands, a drier area is still pretty wet. But we do have a smaller cassette that we can build for, I would think it's probably just a couple of dollars at the most. And within that cassette, we can do only seed germination assays right now. But we do have another, we have designs that will allow us to look at free-floating vegetation. So instead of inserting them into the ground, they float with a little buoyant ring around them, like a little floaty. We have some that we can work with submerged aquatic plants that will actually sink into to a lake or riverbed. We have the ones that we insert into the soil, and then we have this I guess really it's a fourth design, which is a more drastic modification that allows us to do seed germination in terrestrial areas.
Does anybody else have any other questions?
I do have a few more. Now, I know this is more specific, like to experiments that could be done with it. And obviously, if this were a product, you'd let them come up with their own. But I was curious if you've thought about using genetically modified seeds. So perhaps you have, let's say, an Arabidopsis seed that is just the regular Arabidopsis, and then one that maybe has a like an antibiotic resistance, and you were checking to see which one germinates. And if the one that's antibiotic resistant germinates in the water runoff, then you know, okay, well, the other one didn't, this one did. Therefore, I can say that there's likely this antibiotic in this waterway.
Yeah, that's an excellent idea. And because my background is more ecology, our focus has been on native plants, but you're absolutely right. Having something that have plants that have these knockouts or have these sensitivities, but all based on Arabidopsis, which is so well known, is a great idea.
I'm wondering though, I guess now that I said it, there might be regulatory issues with putting out genetically modified seeds right into the, right out in the open like that, in case it were to break.
Yeah, yeah, that's true. And there probably have to be some security built into the design of the FDAs that wouldn't permit that. But having said that, if we look at how much instrumentation is deployed in monitoring our watersheds already, it wouldn't be hard to come up with maybe a stake and a chain to make sure that the FDAs didn't wash away in a flood or something. I think it could be done.
I also wanted to know, what's your current manufacturing process like, or do you have one in place?
Well, at this point, we've manufactured everything ourselves. So the university helped us purchase some of the tools and equipment that we needed to fabricate these FDAs. So we have been doing it. It's not an expensive process. We've deliberately tried to design this using materials that are readily available. Does that answer the question about the?
Yes, it does. I do have one more question, I suppose, about what are you looking to market towards currently? So for example, even if we didn't win this, I'm hoping there's something that you could take away from our presentation for helping you maybe market towards gaining capital or gaining funders. So I was just wondering, who are you currently trying to reach? Are you looking for investors, or are you looking for grants or funds, or are you actually looking to market and bring it to market?
So I can take that, Kevin. So right now we're trying to target grants because there is a little bit of research that needs to be done to demonstrate to potential customers that this can work and can save them a lot of money. So Kevin's done a little bit of work with the giant mine that he talked about in his presentation, but to demonstrate to engineering consultants specifically, I think, on a project that they're currently working on, compare it to standardized testing, would be a really great case to have to then move on and request money from investors and whatnot.
Perfect, thank you.
All right. Is there anything else, Kevin, that you wanted to mention?
Well, I guess a couple of things that I could say is that the responses that we're looking at, and I didn't emphasize those, but things that we look at include root growth and development, shoot growth and development, and these are generally more sensitive characteristics to assess than things like overall biomass. So we have done studies in the past comparing the root shoot morphology, the development of the root and the shoot to more conventional endpoints, and found that they are much more sensitive. So we're confident that this is an approach that is going to show us if there are any contaminants present. It's a fairly sensitive assay. And I guess the other thing I should mention is that, when we talk about this, it's often in regards to monitoring or seeing if there are contaminants and effects on vegetation, detrimental effects. But we should also look at it from the point of view as if we put these out somewhere where we know that there's a quantity or an amount of a contaminant that is supposed to have an effect, and we put our plants out there and find that there's no biological relevance, or there's no impact on the plants that we're working with, we may be able to show that an area is not as problematic as a conventional test may lead one to believe. So it could be a way of freeing up areas or preventing the unnecessary remediation of an area that may not need it.
Anybody else have any other questions? Okay. Okay. So I thought someone came in, but now they left. I was gonna go over some of the slides again, but I think that's it. So the abstract is due January 11th, so you do have a little bit of time to put that together. If any of you have any other questions, feel free to reach out to me. My email is loho@wlu.ca, and I'll be able to answer them or direct them to Kevin to answer. And I guess if that's it, okay, new person. Hi. If you're just joining us, I did have a little, some commercial slides to talk about if you want me to go over. Some of the stuff that we did talk about was a market research study that we conducted back in 2013, and about some of the potential uses that could be done. So that can be used for the device. So just to summarize, the market research study is that they found that site-specific risk assessments was particularly a big market opportunity for the use of this device, and it could save the engineering environmental consulting firms that are currently spending about $100,000 for toxicological testing, down to about $5,000 per site. And that's for in North America, the potential market size is about 5 million. So there wasn't really any direct competitors. So if any of you wanted to know more about the research study, you can also reach out to me, and I can give you a little bit more information about that as well.
Sorry, could you repeat your email just one more time?
Yeah, I can put in the chat box actually, so that.
Oh, that'd be perfect. Thank you.
Is that going to, not to everyone? Oh, okay. I can just send it to you. To send to everyone individually. Okay. Okay. I guess if there isn't anything else, we can close this session. Unless you want to add anything else, Kevin?
No, I'm glad to answer any questions, show any pictures, or provide any more information, whatever you would like. I appreciate you coming in and listening to the talk and participating in the breakout session.
Thank you, everyone. We look forward to seeing some of your pitches, and we're really excited to see what you all come up with. Okay, for anybody who's just entering, we're just about to close session because there weren't any other questions, but if you do have any questions about technology, you can email me, and I'm just gonna send my email out. And I guess we can close the session unless Paulette has any questions.
Oh, no, that's okay. That's great. Thank you.
Okay. All right. Okay. Thanks, everyone.
Thank you.
Okay, bye.